So let's talk about gene therapy. If a person is born with hereditary disease, assume a person born with hereditary disease, means lack of genes or lack of chromosomes. Can a corrective therapy be taken for such a disease? Yes, it became possible. Gene therapy is an attempt to do this. So they attempted a therapy called gene therapy. Why can't we insert the abnormal gene? Why can't we insert in the place of abnormal gene a normal gene? So gene therapy is the collection of methods that allows the correction of gene defects. So if gene is defect, why can't we correct? That has been diagnosed in a child and embryo. So here genes are inserted into the person's cells and tissue to treat the disease. Correction of a gene defect involves delivery of normal gene into the individual or embryo, take over the function of and compensate for the non-functional gene. So the first clinical gene therapy was in 1990. Here it is in 1983 Eli Lilly, here it is 1990. A 4-year-old girl with adenosine deaminase deficiency, ADA deficiency, adenosine deaminase deficiency. So the enzyme is crucial for immune system to function. The disorder is caused due to deletion of the gene for adenosine deaminase. So that 4-year girl doesn't have this adenosine deaminase. In some children, adenosine deaminase deficiency can be cured by bone marrow transplantation. So in others, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. So through injection, we can able to give the functional adenosine deaminase, but we have to give it on a regular basis. But the problem with both of these approaches is that they are not completely curative. As the first step towards the gene therapy, the lymphocytes from the blood of the patients are grown in a culture. So in the first step, what we are doing is we are taking the lymphocytes, are grown in the culture outside the body. A functional ADA or cDNA, usually the retroviral vector, is then introduced into these lymphocytes, which is subsequently returned to the patient. However, as these cells are not immortal, so the patient requires the periodic infusion of such genetically engineered lymphocytes. So, however, if the gene isolated from the bone marrow cells producing ADA is introduced in the cells at early embryonic stages, so this could be a permanent cure, but it was very laborious and risky as well. Now what at present we are doing is we are extracting the lymphocytes and we are introducing the adenosine deaminase production gene into the lymphocytes, and now that will produce the adenosine deaminase, and that causes, so this was conducted for a 4-year girl in 1990. So this is about the gene therapy.